talking about this phenomenon, what is called vegetal rotation. Okay. So we said that the gastrulation that starts in the marginal zone, right? And we are saying, for example, if this is the embryo, right here, and this is kind of the area. So right, these cells right here are going to become the bottle cell, and we are saying when this is going to happen, these cells are going to meet these cells beneath this, and that is going to start the gastrulation. Okay. Now, so we said that cells start involution. Cell involution is not a passive process, right? So what happened that 2 hours before, so 2 hours before the formation of portal cells, what happens? So let me write. So the portal cells have not formed, okay? So this is what we have. No, it's a little bit bigger. Okay, so this is the situation. So your blastocell is there. These are your ectoderm. These are your, this is your animal hemisphere and your vegetal hemisphere. Okay. So we are saying that the cells, these cells are endodermal cells, right? And we are saying this area where the endodermal cell are meeting the ectodermal cell, those cells right here, so these cells which are here on this side as well as on this side, these are the mesodermal cells, right? And how they become mesodermal we'll talk in a minute, okay? But these are the mesodermal cells. Now this is happening before the start of gastrulation, right? So this is, we are saying the gastrulation start when the portal cells are formed, right? And we are saying this is happening 2 hours before the formation of portal cells. So what gastrulation is going to do? Gastrulation is going to allow these cells to actually move in, right? So as these cells are coming, if you remember the picture of the, let me just think about this once more. So if you remember, this is the cleavage. End of the cleavage. Yeah. So you see here, this is the blastopore lip formation and the cells start moving in, right? So if you see here, that means these cells which are moving, they will move in, right? So far it is okay. When those cells move in, when those cells reach here, in this position, they change into mesotome. Up to here, they are not mesotome. But as these cells, they come in contact with the vegetal cell, these cells will change into mesotome. So cell will keep on moving from here to here, here to here. Up to here, they're coming as ectoderm. When those cells touches these vegetal cells, they change into mesotome. That's you have to remember. So they are not mesotome throughout. They are mesotome only at this place where these cells are in contact with the vegetal cells. Okay? So what happens that, again, going there. Okay. So the mesoderm which is here, that mesoderm can be actually have this is two types of mesoderm there. So right here, I will draw it like this. And draw it like this. Okay. So the cells which are here, these are called coda mesoderm. And these are called head mesoderm. Okay. So what happened? This is this is in the blastula stage before the gastrulation, just at the time of mid-blastular transition. So what is happening there? When this is in this stage, this vegetal cells, they start moving actively. So these cells start rotating, and that is called vegetal rotation. These cells in the vegetal hemisphere, they start rotating. They rotate in various different ways. Okay. But what these rotations does, so this is what I'm saying is happening is vegetal rotation. What this does, this phenomenon bring the cells which are at the floor of the vegetal hemisphere. So these are the cells at the floor of vegetal hemisphere. These cells which are at the floor of the vegetal hemisphere, they are actually put right here next to the head mesoderm. They are brought here. Okay. So what that does? So that means they are brought here. So these cells which are brought here, they will, so when these are brought here at this time, they are just reaching here. At that time, the bottle cell formation also takes place. Okay, let me draw it here. So this is the mesoderm here. And right here, the bottle cell formation takes place. And follow this, right here is your head mesoderm, and right there is your coda mesoderm. Okay, so this is your head mesoderm, I'm writing HM, coda mesoderm CM. Now these cells which were here, they are put right there, right? And when these cells are put there, what is happening there? These cells are then called, write it a little bigger, pharyngeal. Pharyngeal, you cannot see it, right? Pharyngeal endoderm. Okay, so these cells are called pharyngeal endoderm. And these pharyngeal endoderm, they are actually going to form the future foregut. Future foregut. These cells are here. Now, what happened? These cells were in the endoderm. They are put here, right there. And these cells, because now the bottle cell have formed, now these were in contact with this, so these cells actually start moving in, right? So what is going to happen? This is going to lead to the third scenario. Let me draw the third scenario here.
Is the pharyngeal just the cell from the floor?
Just a second, let me draw it, then I'll explain it, okay? Just drawing it in different colors, so it's all the colors are more clear here. Okay, now, what was your question?
Is the pharyngeal and
Pharyngeal,
is it just the cell from the floor?
right. Yeah, this group of cells are pushed here. These cells are now next to head mesoderm, okay? Now the bottle cells are formed right here. So these things start moving in along the roof. So that is what is happening. They start moving in, and then they will start move along the roof of the blastocell. Now these cells are now moving in. So this is your pharyngeal endoderm. Then this is your head mesoderm, and this is your coda mesoderm. And then follow this, all these cells are going to turn into the mesoderm. Now, this is the area right here where this thing is happening. So this is the blastopore lip, right? And this is the dorsal, this is the blastopore, and this area is essentially dorsal blastopore lip. So what happened? The order of the involution, how that involution is going to take place, is determined by these cells which were on the floor of the vegetal hemisphere. These come here, sit next to head mesoderm. When they set, immediately the bottle cell form. When bottle cell form, this cell is now in contact with this, so these cells start moving in along with, on top of this pharyngeal endoderm start moving this. So you have the first cell which are moving there. So this zone where cells are moving in, we know that this is called involuting marginal or migrating zone, IMJ. So in this IMJ, in the first three cells, the order of march will be pharyngeal endoderm, which were already here, brought back here, followed by head mesoderm, followed by coda mesoderm. Okay? And then these cells will start moving along as more and more cells are going to come. These cells will start moving along the roof of the blastocele. So this is the roof of the blastocele. Now these are ectoderm. So ectoderm are making the roof of the blastocele, right? So these cells are moving along like this. Making sense? Now look at that thing once more. I believe I have that.
Dr. Morrow,
Uh-huh.
what is this red circle on the left?
This?
Yeah.
Oh yeah, I'm going there.
Okay.
Yeah. So far I'm talking only about dorsal blastopore, what is happening on this side, right? So, let me show you this thing, then we'll go back there and that's going to clear us.
If we look at a cross-section of an embryo of the frog Xenopus, we can see that at this point it is a ball of cells with a
These
fluid-filled
two right
cavity.
there showing there.
The cavity is the blastocele, and the embryo is currently in the blastula stage of development. A blastula contains large yolk-filled cells at the vegetal pole and smaller cells at the animal pole. The three colors represent the three tissue layers that become defined early in embryogenesis. Yellow indicates endoderm, red indicates mesoderm, and blue indicates ectoderm. At the beginning of gastrulation, a few surface cells, called bottle cells, move into the interior of the embryo, followed by other surface cells. We can track the movement of cells into the embryo if we add dye to a few surface cells. The movement of cells into the embryo creates a lip, called the dorsal lip, over which sheets of cells continue to move inside.
Okay. So what we have shown here, dorsal lip so far, right? So we are showing these are moving. Now what is happening? When this process is occurring, the epibole also start, right? It doesn't happen, one happen, then next happen. So what is epibole? Epibole is like these ectoderm cell, they will start dividing, as we said, and they will start intercalating, and they will start moving down. So, when they are going to move down, like these cells, they are going to push this mesoderm more toward this side, because these cells are moving continuously, continuously moving. This mesoderm will be pushed on this side, like it's being pushed here. See, it was here and it's being pushed here. Making sense or no? Now, what is going to happen? See here. Again.
Same time, the ectoderm extends around the embryo surface in a process called epibole. As gastrulation proceeds, a cavity called the arc enteron forms while the blastocele progressively shrinks. The arc enteron is the primitive gut and is completely surrounded by endodermal tissue. The endoderm at the roof of the cavity originated from the outside of the embryo. The cavity is continuous with the outside via the blastopore, which eventually becomes the anus of the animal. As the ectoderm extends around the embryo, another set of bottle cells forms. These
Okay, so what is happening? So this ectoderm is expanding, ectoderm is expanding, ectoderm is expanding, ectoderm is expanding, ectoderm is expanding. As it expands, on the other side here, the new bottle cells are formed. So new bottle cells are formed. From here, this mesoderm, it start migrating in. From here, this migrates. So this is now called, that was dorsal blastopore lip. Now this is called ventral blastopore lip. Making sense? No. So what is happening? We are saying this vegetal rotation take place. Vegetal rotation bring these cells from the floor next to the head mesoderm. These cells are called pharyngeal endoderm. They are going to give you the future foregut of the embryo. Once this is placed there, the bottle cell formation take place, and that's caused involution of these ectodermal cell inside. When these cells are in contact with the vegetal cell, they change into the mesoderm. They have already changed here. So the first cell that enter are pharyngeal endoderm, then head mesoderm, then you have the coda mesoderm. So from here, all the cells are going to start moving in, and this is called dorsal blastopore lip and the blastopore. So these cells will continuously move in. While this thing is happening, the epiboly also start. This epiboly, this ectoderm start extending. This extended ectoderm pushes this mesoderm and everything on this side. And then another set of bottle cells are formed. And from there, these ectoderm will start moving inside, and you're going to have a ventral blastopore lip. So, as these cells are moving in from both the sides, so the cell at involuting marginal zone, they are always changing. They are not the same cell, right? So, in this way, what is happening during this process, what we are getting in the end, as these cells are migrating, we get a new cavity formation. A new cavity formation takes place, and that cavity is called archenterome. Okay? So, then what is there? What is so important here in these structures we are talking? So it is clear or not? So just talk to each other for a second so that we are clear that we are understanding this process.
Is this video on the middle in there?
Hmm?
Is this video on the middle in there?
Can we watch the rest of the video
Oh,
before
yeah,
we
yeah.
talk?
Is the video on the middle though? Like, can we watch it later?
I can put it in the model. Yes,
Yes,
I
please.
can put it on the model. But let's watch it once more.
Seal, and the embryo is currently in the blastula stage of development. A blastula contains large yolk-filled cells at the vegetal pole and smaller cells at the animal pole. The three colors represent the three tissue layers that become defined early in embryogenesis. Yellow indicates endoderm, red indicates mesoderm, and blue indicates ectoderm. At the beginning of gastrulation, a few surface cells, called bottle cells, move into the interior of the embryo, followed by other surface cells. We can track the movement of cells into the embryo if we add dye to a few surface cells. The movement of cells into the embryo creates a lip, called the dorsal lip, over which sheets of cells continue to move inside. At the same time, the ectoderm extends around the embryo surface in a process called epiboly. As gastrulation proceeds, a cavity called the arc enteron forms while the blastocele progressively shrinks. The arc enteron is the primitive gut and is completely surrounded by endodermal tissue. The endoderm at the roof of the cavity originated from the outside of the embryo. The cavity is continuous with the outside via the blastopore, which eventually becomes the anus of the animal. As the ectoderm extends around the embryo, another set of bottle cells forms. These cells migrate into the embryo, and other surface cells follow them, creating the ventral lip of the blastopore. By the end of gastrulation, the ectoderm has surrounded the embryo. Endoderm lines the inside, and mesoderm lies between the two. Additionally, the fates of specific regions have become determined. The endoderm gives rise to the digestive and respiratory tracts and associated structures. The mesoderm gives rise to the skeleton, circulatory system, muscles, excretory system, and most of the reproductive system. The ectoderm gives rise to the skin, sense organs, and nervous system.
Okay, I'll put this on that, but talk about this. I want you to talk about, that's a process you can visualize, but I want you to talk about the vegetal rotation. What is happening in vegetal rotation? What are these type of cells? What is happening? What is the order of the march? Because this is important for the next thing what we want to talk about. So talk to each other for a minute, then we'll go further.
I don't like the way it reads, since it doesn't really show it. I read it like this, and I'm gonna like take my colors. So this becomes basically like this pushes the little plastocele thing down here, and this brings with it a cavity from the lip. That's called the entrance cell.
Cells move from the lumbar pole basically to the mesoderm.
And then these move in and form, like these dorsal ones, they like reach over here, they form an internal little
They change to mesoderm.
at the ventral.
And then cells in the visual hemisphere start their rotation and it brings
I'm gonna draw
the cell at the floor of the visual hemisphere that's next to bottom cells form. And then
Yeah, I'm just gonna.
that's when you have this formation. So we come up here, rotating.
I'm assuming do they connect,
And then
like the other
they
one
change to
comes
the
up and.
pharyngoendoderm
I don't
once
know,
they
you
meet
look
the
like
head mesoderm. And then this allows the cells
I mean,
to
the
start
mesoderm
moving in, and then
Like mentally they
and then
connect, right?
pushes up
What?
the
No, these are the where they come from,
endoderm,
like
and then the head
this
mesoderm.
one comes all the way up to here, and then like that one sits like right there. They don't touch.
Or that could be
They don't ever meet, like the mesoderm,
Do the
Say again?
meet?
the mesoderm, the little, this
Uh-huh.
part.
Do they
It came
meet?
all the way up here and sat there, and then it made another like.
Yes, yes, it move in.
Yeah,
It also, so it's coming here, so this whole thing also start moving like
and
this.
then it makes a new cavity.
Oh, no, that was kind of like not drawing properly, it's a one cavity, they're kind of like showing it like this, it's a one cavity. So in that video, they're trying to show another cavity, there's no another cavity.
So there's like a little like
Yeah.
space right here though, between those two, right?
Yes,
Okay.
yeah. So essentially, there is one cavity which is formed here. So the whole thing will have one cavity, it will be like this, let me draw it.
Plastic orbit at the blastophore, which is confusing. So, yeah, and as the ectoderm expands, it pushes the mesoderm in, and then that forms the red part.
I honestly kind of consider it an inevitability after this because you have to draw it every time and like you put your notes in.
Mesoderm,
And everybody
and
sees
then
their
mesoderm.
notes.
Yes. So that's really essentially this look. That's determine this type of things. The architecture. Yeah.
So it will be like this in the end. So this will be archenteron. Archenteron. Endoderm. This will be, the red will be your mesoderm. This whole thing will be a mesoderm. And then you will have your ectoderm. It will be like that. So that small streak which is shown there in the video, that's not right.
Okay.
Okay.
I don't think it's called seal building and not epibody.
So this will be the end product. You will have a big new blastocell will be gone. The new cavity archenterone is going to form. Archenterone surrounded by endoderm, then mesoderm, and then ectoderm.
So how does the blastocell like disappear?
Hmm?
How does the blastocell like it just gets
Oh,
filled?
because if this is there, the other cells are entering.
Okay, so it just gets filled.
Yeah, it just filled with the other cells. And if I was drawing it right, I should have shown this thing right here. I should have started this thing from here. It should have been like this, right? Because this is happening at the dorsal side. It's happening here, right? I should have tilted like this.
Wait, what? Say that again?
So I should have tilted this thing like this, you know? This thing should be here, should be like
Oh,
this.
okay.
So tilt it at 90 degree, I will say. Draw this like this.
So that's basically that,
Yeah.
That'd still be correct though if we like just drew it like this on the test, for example, to like describe the end product.
This is the end product. This is, I was telling you, the generalized scheme.
Okay.
This is how it will be.
Okay.
So that's what is happening in the end. You essentially the same thing, but that is more of a generalized scheme, right? Any question here, anything that is doubtful? You're okay over here? You're okay? Okay, now.
So is that little like space still the dorsal lip, or is like that where like the dorsal and ventral lip?
Say it again?
That little like this little space, is that like the dorsal and ventral lip? Like where
Yeah,
the veins-
yeah. So this is the ventral lip, this is the dorsal lip. The lower one is the ventral lip, upper one is the dorsal lip. The pore is a blastopore, but it's the future anus of the embryo.
Okay.
Okay. Now what is happening here? So let me draw that thing once more so that so I'm just drawing this thing here. So I'm saying these cells have moved in. And then okay. So these are the first three cells, three type of cells which were moving in, okay? So these cells which are here, the pharyngeal endoderm, they are going to give you prospective foregut endoderm, so essentially going to make the foregut. What they do, these cells, the pharyngeal endoderm, that actually express HHX, HHEX. These are the genes which they express, okay? Set of genes. And these are critical for head and heart formation. HHEX genes, so they are expressed in pharyngeal endoderm, but their product is responsible for formation of head and heart. Okay. So after this, you have So this is moving here. So this is this area, the first area. This is called head mesoderm. Okay, so head mesoderm is also called precaudal plate. It's also called precaudal plate, and that is actually making a gene called goosequoid genes. Group of genes called goosequoid genes. These genes are responsible for head mesoderm formation. So this area is, as the name suggests, is going to give you the head mesoderm, right? So you will have the head from there. And then what we are going to have is this area, the third one. This is called coda mesoderm. This expresses the gene called brachyury, also called XBRA1 genes. This is going to give those genes. Brachyury gene, also called XBRA gene. So these XBRA genes are brachyury gene. Now, this part, coda mesoderm, is going to give you a transient structure. That transient structure is called notochord. This notochord is a transient structure. That means it is present only in the embryo, right? But this is responsible for the formation of nervous system. So all the nervous system that is going to form because of this mesoderm which is there. So that's why we are saying that's why we're specifically talking about these three, because these three are critical. Otherwise, if you remove these from there, what is going to happen? No nervous system is going to form. Okay, so these first three are critical. Now, as they are moving from here to here, they will ultimately reach here. We said that sperm entry make dorsal ventral polarity, right? The end of the gastrulation, because this is going to give you the head, right? So the anterior posterior head tail polarity is determined by the migration of these cells or mesodermal cells which are going to move during the gastrulation. So we get anterior posterior polarity by the end of gastrulation. Making sense so far? Now, there is another thing happening. We are saying that epiboly is taking place, right? So if we are saying, just let me draw it in the other color. If you're saying like you have the other cells here, so these cells are migrating, right? Now these cells are migrating also. These cells are migrating on the roof of the blastocele, right? So when the cell migrates, cell cannot migrate on its own. You need to have some sort of cell adhesion protein rods on which these cells can migrate. So what happened? The cell, the ectodermal cell, on the roof of the blastocele secrete fibronectin. And fibronectin fibrils are made on the base of the ectodermal cell on the roof of blastocele. So what is happening? The cells which are here, they are secreting fibronectin right there. So this fibronectin is secreted. Fibronectin, so fibronectin, these are the fibronectin fibrils. So fibronectin is secreted by these ectodermal cells. They make fibronectin fibril. So kind of make a path on which these cells can migrate. So if somehow you destroy this fibronectin, this migration is not going to take place. Making sense? Or no? Okay. So talk about this for a second, then we'll go to next topic. So what we are saying overall, we are saying that these cells which are moving in, these mesodermal cells, they determine the anterior posterior polarity of the future embryo. We are saying the pharyngeal endoderm, which are going to make endoderm for foregut, they have expressed the gene HHEX. Those are the set of genes, those are required for head and heart formation. Following those, we have that head mesoderm, also called precaudal plate, that express Goose-Quiet genes, and those Goose-Quiet genes are responsible for formation of the head mesoderm. So the head is determined right there. Then we are saying we have these later the coda mesoderm. The coda mesoderm is also called caudal plate. Okay, so the coda mesoderm is there. Coda mesoderm express brachyuria XBRA1 gene, and these genes are critical for the induction of the nervous system from the ectoderm. And we said this coda mesoderm is going to form a transient structure called notochord that is only present in the embryo. But this structure is responsible for changing the ectoderm above it into the nervous system. So think about it. So this is if you remember that I showed you that we made that fate map, and we showed right here the nervous system, right? We showed right here the nervous system because right here is the coda mesoderm. Coda mesoderm is going to make notochord, and the ectoderm above it is going to become the nervous system. Making sense? So then we said the movement of this cell along the roof of the blastocele is determined by the fibronectin, which is secreted by these ectodermal cells. They make fibril, kind of the roads on which these cells can actually travel. So if you destroy this fibronectin, you will not have any gastrulation. So embryo will just stop right there. Making sense? Okay, so talk for each other with each other, and we'll go further.
So this one's in the fucking red one. And then the ectoderm above it becomes a root? Yeah. So you said, Dr. Bohr,
Hmm?
so you said the quarter mesoderm, after it expresses that gene, gives the notochord.
Yes,
And then the ectoderm cells above that, like it forms those into the notochord?
yes, ectoderm above that is going to change into nervous system. Above the notochord. And we'll talk more when we talk about how the nervous system is formed, but that's what we have to remember here. This
So
thing.
does like the notochord just disappear then?
Hmm?
Does the notochord just disappear then?
Yes, after that notochord, it disappears. Notochord is only in the embryo. In some lower organism, notochord persists, but not in mammals and vertebrate. It just goes away.
Doesn't that move throughout the whole thing?
Yeah.
Yeah, but this is still not embryos. So one's like all different.
So for these
then, like,
these cells, you said they're.
No, but they're the group of cells.
Yeah. the group
Not three
of
cells,
man.
they're like thousands of cells.
Right.
Right?
Their migration determines the polarity, is that?
Yes, they do, because you have they are coming here, so they are going to, this is head mesoderm, right?
Yeah.
Pharyngeal endoderm. But no, this is head mesoderm. So if they are moving on this side, that means this side is going to the head, right? So they are defining head there. The first head formation, then slowly head. And that's what we show in the previous figure, which is right? From here they are reaching up to here. So head and tail. Making sense?
Yeah.
Any doubt here, William, it is okay?
I'm good.
You guys are okay here? Okay, now. We are on the last topic, and that is specification of the germ layers. Remember, we are saying that mesoderm is formed, endoderm is formed, ectoderm is formed, but how they are formed, right? So that's what we want to talk about. Think about this structure again. And we are saying this is endoderm, mesoderm, ectoderm. Okay, now what is happening? So we are saying that this specification of germ layer actually happens because this is the start of the gastrulation, right? Gastrulation is making 3 layers. But at the beginning of the gastrulation, we already had mesoderm, ectoderm, and endoderm. We are saying these are formed like this. So that means that the specification of the germline cells is already determined in the blastula. And in blastula, we said there is a stage what we call mid-blastula transition, where we said multiple genes start expressing, right? So what happened right before the mid-blastula transition, right before that. So in this part, in the vegetal portion, in the vegetal hemisphere, the cortex. Now, this is actually right there in the egg. When the egg is there, remember when we're talking about the egg structure, we said egg has differentiation factor. Remember when we're talking about egg structure, we're saying though differentiation factors are distributed, they are not evenly distributed. They are distributed unevenly. So in the egg itself, right here to the cortex, so this is the egg, this is the animal hemisphere, this is the vegetal hemisphere. Right here to the cortical cytoplasm. This is the cortical cytoplasm. In that area, the wedge T mRNA is present. So throughout here, wedge T mRNA is present in these cells. Not in the cell when it is just one cell, right? In the egg. Now we said it divide. When it divide, giving you various cells, so each of this cell will have wedge T. Making sense? So all those cells which are here in the vesical hemisphere, they have wedge T mRNA. Now wedge T mRNA, what it does? Wedge T mRNA activates set of genes just at the beginning, prior to mid-blastula transition. Okay, now what it does. In the cells of vegetal hemisphere, because this is present only in these cells, all those cells which are here, all these cells, they have wedge T mRNA, right? So wedge T mRNA, what it does is start its translation. So you have a wedge T protein made, and wedge T is a transcription factor. Okay, so what it does, it activates, so transcription factor can bind to promoter and basically start the translation, right? So what it does, it activates SOX17 transcription. Excuse me. So SOX17 start baking, and what SOX17 is, SOX17 is a transcription factor. And what SOX17 does, SOX17 activate a set of genes for endoderm specification. So now this cell, because now they're having wedge T activity, wedge T activates SOX17, SOX17 activate endodermal gene. So this whole thing has become endoderm. That's the first thing that's happening. Then what happens? Then what happened, that this wedge T, once these cells have become endoderm, first step is SOX17 activate endodermal gene, this become endoderm. Now what it does, wedge T in the endodermal cell induces the formation of a paracrine factor and that paracrine factor is called nodal, NODAL. Okay, now once this nodal is there, now this nodal is being made. Let's say these cells are making nodal. So this is nodal. Okay, paracrine factor means it's going to neighboring cells, right? So this paracrine factor which is secreted here here, it goes to the cells above it. So these cells are still not red, they're still not mesoderm, they're still ectoderm, right? So let me draw it in the beginning. So these cells have changed into endoderm. Now these cells are still ectoderm, still not become mesoderm. So this nodal go into the cells above it. And what it does, when it goes above them, nodal activate phosphorylation. So nodal is going up, and right here nodal going, and there is protein called SMAD2. In the cells above it. So in the cell above it, now these cells, nodal going up, and these cells start making phosphosmad2. Okay. So what happened that phosphorylated SMAD, what it does? Phosphorylated SMAD activates, let me draw other color. It activates the expression of number 1 EO mesodermin. EO mesodermin. And brachyury. Brachyury is again extra. These two genes. Okay. And when these two genes are made, EO mesodermin and brachyury, that activate genes for mesoderm specification. So these cells which are right there, these cells, they changed into the mesoderm. So what is okay? Now, so they have phosphorylated SMAD2 and EO mesodermin. So phosphorylated SMAD2 goes up there, tell them to activate EO mesodermin and brachyury. Okay? Now, EO mesodermin and SMAD2, phosphorylated SMAD2, together. Now this is interesting. VEGT secrete nodal. Nodal tell these cells to make phosphorylated SMAD2. SMAD2 tell make EO mesodermin and brachyury. Right? Now EO mesodermin and SMAD2, they were right here. They come down to the endodermal cell. And what they do, they together activate zygotic VEGT. So this is a positive feedback. So then it is making more of the VEGT, cells are dividing more VEGT, so you have enough endoderm, and this VEGT is going to go back and tell them make more nodal, make more phosphosmad, make more eodermin and extra, right? So then there's a positive feedback loop. So it's continuously they are inducing each other. So far it's okay. Now, there is one more thing. If I draw it like this, let me just draw it like this. So let's try to just sum this thing up. So we are having wedge T, right? Wedge T, then wedge T, what wedge T is doing? Wedge T is telling this cell here, activate SOX 17, right? When the SOX 17 is activated, this cell become endoderm. Now then this wedge T is inducing nodal, right? This nodal is going up, producing phospholipid SMAD2, both sides. Okay, then that is doing two things. EOM, I'm writing EOM as isomerism and XPRA1 in both. EOM and XPRA1 in both. Now this eomisgermin and SMAD2 together, they're coming down and they're inducing more wedge T, right? But what happens on this side, on the dorsal side? Now this is the dorsal, this is the ventral. On dorsal side, there is an additional so there's an additional what we call VG1 mRNA is there. Additional VG1 mRNA is there. That VG1 mRNA from here goes up. Okay, and when it goes up, it actually inhibits Wnt. Okay, so now that's important thing here. We had the mesoderm on this side, mesoderm on this side. This mesoderm was specifically making precaudal plate or head mesoderm and coda mesoderm. But this side was not making that, right? So that mesoderm here and mesoderm here is different. And that is different because of the VG1. VG1 goes from here to here and inhibit Wnt and make this mesoderm different than this mesoderm. Let me go back and repeat everything so that everything is clear. We are saying that specification of germ layer that start in the blastula. Just before mid-blastula transition, wedge T, which is a transcription factor present in the vestal hemisphere, that starts expressing, start making protein. You have the wedge T protein there. And once wedge T is there, what that does in the vestal hemisphere, in this cell that activates SOX17, and SOX17 is transcription factor that activates the endodermal gene, and this all can become endoderm. Then what wedge T does, wedge T tells these cells to make a paracrine factor called nodal. Nodal goes to the cell above them and tell those cells to phosphorylate SMAD. When SMAD2 is phosphorylated, that tells those cells to make eomesodermin and brachyury gene activation. Once those two genes are activated, those transcription factor, they activate series of genes that are going to make this particular structure as a mesoderm. Then we are saying that eomesodermin and phosphorylated SMAD2, they together can activate wedge T in the endoderm so that you have a positive feedback loop. You have continuous endoderm formation, continuous mesodermal formation, because it's a continuous process, right? As the cells are moving in, they are changing into mesoderm. Then we are saying on the dorsal side, there is another mRNA called VG1 mRNA. That mRNA is factors goes up, and when it goes up, it inhibits Wnt signaling. And we are saying that is critical because this side of mesothelin is different than this side of mesothelin. Now, what we have not talked at all about ectoderm. What happened to ectoderm? Right? We are saying ectoderm changed into skin and the nervous system. That signaling does not come from here. That signaling is going to come from mesothelin. We'll talk later. But if you knock down VEGT, if you remove VEGT from here, what is going to happen? The whole cell is going to become epidermis. Nothing, whole embryo will be skin. Nothing else is going to form. So VEGT is the critical, is throughout, is critical for what we are. So talk for this about a minute, then we are done. And we will have our exam on Monday on this topic, amphibian early development and in vitro fertilization. Are we out of time or we?
We're a little over.
Little over? Oh,
It's 10:15.
oh.
It's alright.
Yeah, but we had to finish this, that's all. So we will be meeting in the lab at 12:30?
Yeah, they'll be there at 12:30 and we're gonna come after at 12:30 class.
So we'll be using the same, we'll be making those the 24 well plate, right? And we will be doing the same thing. Take out those neosphere, put trips in there, count there, and then we will have. So you want me to draw it if you have 5 minutes? So that we are on the right track, at least the